Hello, I'm Angelica Ronald and I'm the Director of the Genes Environment Lifespan Laboratory at Birkbeck within the Department of Psychological Sciences. I'm really pleased to be doing this video as part of the Birkbeck Inspires series. I'm going to talk a bit about the research in genetics we do at Birkbeck in human genetics, and also tell you a bit about our celebrations this year of Rosalind Franklin's work. So just to give you a sense of where we are, well, at the moment, obviously, we're working remotely because of the lockdown. But previously, and hopefully, obviously, in the future, we will be back at Birkbeck in WC1. So here's a picture of the lovely square by Birkbeck and some pictures of my team. So I co-direct the lab with Dr. Emma Meeburn. And here are some of our present PhD students and postdocs. So the GEL lab is really focused on conducting research on human genetics, and particularly focused on the genetic and environmental risk factors and influences on infancy and child and adolescent development. So we study a range of phenotypes, including autism and ADHD, infant traits, and also adolescent difficulties such as mental health problems. We're also really fortunate to have this wet lab where we can do additional genetic analyses and also store samples in a biobank. And we have a variety of research themes within our group. You can see here from this word cloud the kind of themes that we're interested in, and a list here in our website can be found of some of our current and active projects, as well as some further ones that are not listed here. So we're quite busy and conducting a variety of exciting research. But I'll come back to a bit more about the GEL lab research towards the end of the talk, but I thought now I'd tell you a little bit about the anniversary we're celebrating this year, which is the 100th anniversary of Rosalind Franklin's birth. And the reason we're doing that is that Rosalind Franklin is really a pioneer in terms of science, and particularly we are very indebted to her within the world of genetics for the research she did in her career. So she was born 100 years ago in London, and she did a variety of different research. She worked in the University of Cambridge and also King's College London and Birkbeck. But first of all, I'll talk you through some of the events that we've been doing to celebrate her birth, and then I'll go a bit more into detail about herself. So one thing we did earlier in the year, before the lockdown obviously, was to run a variety of very busy and fun workshops with primary school children, so 10 to 11-year-olds, and they learnt to extract DNA from strawberries, and they also attended a sort of Genetics 101 workshop with us all. So here are some photos of our team at that event, and we worked with well over 100 children, so that was a lot of fun. And then more recently, since the lockdown, obviously we've been thinking of things to do that can be done remotely. And one thing we've done is started up a blog post, a blog series. So in our first one, our PhD student Chloe Austerberry talks about her research that she's doing during her PhD, but she also reflects on what it's like to be a female genetics PhD student 100 years after Rosalind Franklin's birth, and thankfully some of the improvements in science in that sense. So who is Rosalind Franklin? So she was a chemist and a X-ray crystallographer, and we're celebrating her because she made some really critical contributions to the understanding of the molecular structure of DNA. And that sounds like a bit of a sort of complicated thing to say, but in essence, her work really helped us understand how DNA copies itself, and that's very important for understanding how things are inherited down generations. And I also thought I'd mention that she's someone who had quite a diverse scientific career, which also inspires me. So she worked not only on DNA, but also on RNA and on coal and on graphite, and also on something that's extremely relevant to us all at the moment, which is viruses. So she worked on the molecular structure of viruses while she was at Birkbeck, so hence she's a Birkbeck alumni. But if we just zoom in on her work in the field of genetics, and why, as geneticists, we're celebrating her history and her birth, one of the key things she did, and this is just one example of her work I'm going to show you. This is an image called Photo 51. It's thought to have taken her around a year to develop using this method called X-ray diffraction, which is quite a complicated method, but what happens is that the way that this image turns out using X-ray diffraction reveals to the scientists the shape of a molecule and therefore its possible function. And there's quite a bit of a story relating to this photo 51, because it is understood that a male colleague of hers actually removed this photo from her desk without her permission and showed it to some scientists you may recognise possibly. He showed it to James Watson and Francis Crick, and they used this photo together with other work they had done in order to describe the structure of DNA, deoxyribonucleic acid. And that led on to them winning the Nobel Prize. Unfortunately, Rosalind Franklin was not really celebrated in this way. She had also died before this Nobel Prize was given, and she's often not credited for the amount of work she did that contributed to this scientific finding, which is another reason why we're celebrating her now. So just a little bit of science to fill in before we end. If you're not familiar with genetics, DNA stands for deoxyribonucleic acid, and it's a molecule we find in nearly every single cell in the body, and it's very good at copying itself, which is how cells replicate, both in terms of inside your body and in terms of creating offspring. So we can start to study in genetics what causes individual differences between people, and that's something that the GEL lab is interested in. So in this image you can see lots and lots of black letters and a few red ones. So what that's representing is the fact that in your DNA code, which is made up of letters, base pairs that are represented by scientists as letters, most of the code is exactly the same between all people. But in a few instances in the genome, you get variation between people, and this is what we think causes some of the inherited differences between people. These are represented by the red letters in this diagram, and so looking at these variations in the genome, we can start to try and understand some of the inherited differences caused. Genetic influences that cause differences in things like eye colour and height, but also perhaps more complicated phenotypes like hyperactivity or mental health issues that work in combination with the environment to influence our likelihood of developing a particular phenotype or trait. It's also human genetics. I've really included this slide to emphasise the fact that human genetics is a massive field and influences a wide variety of things, both applied and academic. One thing I lecture in my undergraduate courses at Birkbeck is on evolution, and it's an interesting topic to think about the consumption of milk. So we think that through human genetics we've been able to understand that up till about 3,000 to 5,000 years ago, all humans stopped being able to digest milk after infancy. But around 3,000 to 5,000 years ago, we believe that the genome evolved in some people so that they could digest milk. And that's why some people in the world can digest milk as adults and other people can't. There are variations, and we think this is due to evolution. So that's just a touch on a different angle in which human genetics is useful, and obviously we have a lot to thank Rosalind Franklin for. But cycling back to the GEL lab work, I just thought I'd mention a handful of things that we've been doing in the last year. So one paper we've published this year is led by my PhD student Vikas Barkhuizen, who just got his PhD at the end of last year, and he's now a postdoc in our lab. I'm really proud of this work here, published in Translational Psychiatry, which is a peer-reviewed journal, because he shows novel findings about how adolescent psychotic experiences, which are a type of mental health problem, may link to later psychiatric disorders in adulthood. And he also uses some of the very latest state-of-the-art methods in our field that have just been developed over the last 5 years. In addition, I thought I'd mention an editorial that I wrote just this month in the Journal of Child Psychology and Psychiatry, which tackles some of the strengths, weaknesses, opportunities and threats that polygenic scores can bring to child and adolescent psychiatry. And finally, just to emphasise that we're always trying to find other ways to communicate about our research. And this link is a link to a podcast in which the Association for Child and Adolescent Mental Health interviewed me this spring as well. So again, just trying to find ways in which we can talk about our research at Birkbeck through a variety of channels. Thank you so much for listening, and please find out more by visiting the Birkbeck website, if you so please. Thank you.